have Sarah Kuppert. She was born and raised in Germany, where she got a bachelor's degree in English, business, and economics. She's focused her research on linguistics and specifically animal communication, and ended up switching directions and started her master's in environmental science and policy in the spring of 2013. Her research focus is on conservation genetics and stream local salamanders. The quote that she's chosen for tonight: "The most successful scientist thinks like a poet, wide-ranging, sometimes fantastical, and works like a bookkeeper. It is the latter role that the world sees." Edward O. Wilson from The Meaning of Human Existence. Can we please welcome Sarah to the stage?
I didn't expect so many people here. What about this one? Hello? All right, I'm gonna put it a little closer. All right, everybody, my name is Sarah Kuppert, and I am a master's student of environmental science and policy here at George Mason, and my focus is on conservation, and particularly on conservation genetics. And what I do with that is I detect the species presence with molecular methods in stream water. So ever since I was a small child, I was really into animals. And I sure loved my ponies and fluffy kittens. But what people didn't tell me when I started going into conservation is that it has to do a lot with this, poop. That's what it all comes down to. So you probably now wonder, what does all of this have to do with poop, conservation genetics, salamanders? So I will tell you. So when you introduce yourself as a conservationist, a lot of people think of you as a tree hugger, which is totally not true. However, people also think that you should drive a green car, which I do. Well, who can say no to 50 miles per gallon, right? They also think that you cuddle a lot with cute and fluffy animals. But the reality is not that. Again, it comes all down to this, shoveling poop. So I also consider myself as a herpetologist, and herpetology is the study of reptiles and amphibians. So a lot of people get really scared when I first start talking about my research animals. A lot of people ask me, herpetology, do you study herpes? Or a lot of people also ask me whether I do stuff like this, like Steve Irwin, the crocodile wrestler. When I tell people that I work in genetics, the reaction is quite a bit different, and they usually think that I'm really smart. I like that. However, it always comes back to this. Science is poop, let's face it. So this is just a short introduction now. Let me start with my real research topic. It's not that funny, unfortunately. So the whole reason why we study conservation is because of this. Large biodiversity declines. So nowadays, 1 out of every 3 amphibians is threatened with extinction, and a lot of bird species, a lot of mammals, sea turtles are heavily hit with extinction risk, and especially fish are also on the list. So why is that? It's because of us humans. So we cause a lot of habitat destruction, pollution, and we bring in invasive species like, for example, zebra mussels and lionfish, who just take up resources from local species and drive them into extinction. And then last but not least, also climate change is a major risk. So nowadays, the managers and climate conservationists have to do quite a bit of work in order to keep those species healthy and stop them from further decline, and do that by using adaptive management. So this is a circular process, and it starts with the monitoring of the species. So you go out and you look for the species, you see how many there are, and then you note over the years whether there's actually a change, declines, potential diseases, or anything like that. And then you use that information in order to integrate policies and regulations so you can plan for the future and implement changes in order to keep the species from decline further. And all of that while doing the monitoring and repeating the whole circle. So, however, for some species, it is particularly difficult to monitor them, like my research organisms, the two-lined salamanders. So they're relatively small in size. They're very, they like to hide a lot under logs in fast-flowing streams that sometimes are very difficult to access, and finding them takes a lot of effort and a lot of sweat. And so they're also active only at nighttime, which that is a major problem for researchers that are trying to find them during the day. So in order to monitor those species better that are very cryptic and very small, we came up with a new approach in order to monitor species in the environment without even ever having to see the animals. And that is what I study. It's called environmental DNA, for short eDNA. So what you do here is you take water out of the environment or soil, and you detect the species presence with molecular methods. So can anybody guess what this is for? Yeah, I know you're all in college, so you know that, right? However, it is not what you think. So this is the amount of water that we need to detect the full biodiversity in a pond. So that's quite impressive. You only really need 15 milliliters to really see however many species are in that environment. However, if you monitor streams like I do, it's quite a bit more. It's between 1 and 5 liters. So the question is, what is the DNA that we get out of the water? And here it comes back to this. It's the poop, majorly. It's also some of the skin cells that are sloughed off, specifically for amphibians, but it is mostly poop. So what do I do in order to get that out of the water? First of all, I filter the water. So I go into the field and I have this nice little filtering equipment, and it contains a small filter that has 0.2 micrometers of a pore size, so very small particles get stuck there. Then I go into the lab and I do the extraction. So this involves a lot of components, and you do a lot of washing, a lot of spinning, and you add different chemicals. And in the end, you end up with this tiny little bit of clear liquid here, which is your pure DNA. So that's all the DNA that was in your sample. And then the next step in the process is something really cool. It is called polymerase chain reaction. So this polymerase chain reaction is basically you copy and paste the DNA fragment that you want to amplify, and you do that by putting it into a machine that has 3 steps. The first is denaturation, and it is at 94 degrees. So the double helix of the DNA gets unrolled. Then in the step 2, the annealing procedure at 54 degrees, you bring in some primers that you designed to fit your species DNA fragment that you want to amplify. Then that docks on and kind of goes along the strands and copies whatever there is in the 3rd step, the extension. So you have all those small little base pairs of DNA floating around, and the primers grab them and kind of extend it and form 2 more double helixes. And this step gets repeated between 40 and 50 times for my method. So in the end, you end up with millions of copies of the same piece of DNA that you want to amplify. Then the next step after this is called gel electrophoresis. So you have a squiggly gel that you put into a chamber that is full of a solution, and you load your DNA with a dye into those wells. Then, because DNA is electronegative, you switch on the light switch, and the DNA migrates from the negative to the positive electrode because it gets attracted through it. So the DNA gets sorted by size that way. You know in advance what kind of size you're amplifying. So in the end, if you look at your gel and you look at the bright bands on the gel, as you can see here, you can see whatever size your DNA is. So also one thing you should know about the DNA fragments is the smaller sizes of the DNA fragments, they travel faster through the gel, so they end up all the way at the bottom. So this is how I amplify the DNA out of the water, and this is how I could possibly save the world with poop.
All right. Yeah, you are allowed to ask questions, so.
I'm sorry for coming.
It's all right.
Me knows I asked way too many questions.
Sure.
I was just wondering how you quantify. So you can, using PCR, you'll be
Yes.
able to detect that the salamander DNA is there among the many other different species. How do you figure out how many salamanders that represents?
That is something that I cannot detect with this method. It's solely for species presence or absence. If you want to detect how many species there are, you will have to use a different method called qPCR, and this is for the quantification of the actual DNA, and you see how many copies there are in there.
Thank you.
All right, let's hear it again for Sarah Kuppert.